Welcome to this explainer. Today, we are diving straight into a biological mechanism that is so revolutionary, it literally spawned a billion-dollar medical industry. We're talking about a discovery that earned one of the fastest Nobel Prizes in history. So, let's figure out exactly how our cells manage to hit the brakes and silence their own genes.
To really wrap our heads around this, I want you to imagine a busy cell as a massive, bustling factory. Inside this factory, you've got these assembly lines running constantly, churning out all the necessary products that keep you alive. But suddenly, the factory gets a signal. It needs to halt just one malfunctioning assembly line, but without shutting down the entire factory floor. How on earth does it pull off that kind of logistical nightmare?
Well, let's start with Section 1, inside the cellular factory, DNA and mRNA. Okay, let's map out our factory floor. First up, we have DNA. Think of DNA as the master blueprint locked safely away inside the control room, or the nucleus. It is way too valuable to risk damaging. So instead of using the original, the cell dispatches temporary copies, called messenger RNA or mRNA, out onto the factory floor to act as working instructions.
And once those mRNA instructions actually hit the factory floor, they're used to build proteins. Now, these proteins aren't just byproducts. They are the literal building blocks, the critical machines, and the enzymes that keep the whole cellular factory humming along day in and day out. Every single physical trait and chemical reaction in your body relies on these proteins being built perfectly from those mRNA blueprints.
Which brings us to Section 2, the molecular mute button, introducing RNAi. So going back to our factory problem, what if a protein malfunctions? Or worse, what if a virus tries to hijack the factory for its own use? The cell desperately needs a way to stop it. Enter RNA interference, or RNAi. This is the molecular mute button. It's a completely natural biological process where the cell uses small RNA molecules to physically block or just utterly destroy those temporary messenger RNA copies we just talked about. If the mRNA is destroyed before the factory workers can read it, the protein never gets made. The gene is effectively muted.
Now, you might be thinking, we've known about this forever, right? Actually, no. This is a relatively recent breakthrough. It was discovered by scientists Andrew Fire and Craig Mello in these tiny C. elegans worms. And the discovery was so profound, so fundamental to our understanding of biology, that they won the Nobel Prize in Physiology or Medicine in 2006. That was just 8 years after their discovery. In the science world, that is lightning fast.
Let's dig into the exact mechanics in Section 3, Dicer, the molecular scissors.
Let's look at the step-by-step of how a cell triggers this intense silencing response. The whole thing kicks off like a microscopic security alarm when the cell detects double-stranded RNA. Normally, RNA is single-stranded, so whether it's coming from a virus, a rogue genetic element, or even a scientist's experiment, the cell instantly spots this double-stranded RNA and flags it as a massive threat. To neutralize this threat, the cell deploys Dicer. And I love this name because it does exactly what it sounds like. Dicer is a highly specialized enzyme that acts like a pair of molecular scissors. It hunts down that unusual double-stranded RNA and starts chopping it up before it can do any damage. Dicer meticulously snips that invading RNA into tiny, manageable fragments. And I mean tiny. They are incredibly precise, usually exactly 21 to 25 nucleotides long. Once they're chopped down to the specific size, we give them a new name, small interfering RNA, or siRNA for short.
Moving right along to Section 4, RISC, the security team.
Okay, now that the intruder has been diced up into these siRNA fragments, let's track the rest of the timeline. Step 1, the double-stranded RNA intruder enters. Step 2, Dicer cuts it into siRNA. Step 3, the cell's elite security team steps in. The siRNA joins a specialized protein complex called RISC. That stands for RNA-induced silencing complex. And finally, Step 4, RISC uses those fragments to hunt down and destroy matching mRNA. This part is just absolutely fascinating. Once the siRNA is inside that RISC security team, RISC deliberately separates the two strands of the siRNA. It literally tosses one strand away, just discards it, but it keeps the remaining strand, which we call the guide strand. RISC uses this guide strand as a literal search key, endlessly scanning the entire cell to find any natural messenger RNA that perfectly matches it.
When you compare a normal sequence to an RNAi sequence, the contrast is stark. Normally, mRNA floats freely, gets read, and produces proteins. But under RNAi silencing, once RISC binds to a matching mRNA, game over. The target is either shredded into pieces or its ability to be read is permanently blocked. It's simple, Brutal cellular logic. If there's no mRNA, there's no protein. The gene is totally silenced.
So, what's the big picture here? Let's get into Section 5, the power of silence. Harnessing this molecular mute button is profoundly revolutionary, mainly for 3 reasons. First, it's a built-in shield. It actively hunts down viral RNA to protect us from the inside out. Second, it's an internal regulator that naturally switches off our own genes when we don't need them. And third, it has become the ultimate biotechnology tool. Scientists can now intentionally silence specific genes just to figure out what they actually do.
And building on that, the medical impact is just staggering. Researchers aren't just studying RNA anymore, they're actively weaponizing it to heal people. We're talking about cutting-edge RNA-based therapies. By deliberately targeting the harmful genes responsible for viral infections, cancers, and complex genetic disorders, scientists can literally tell the cell's own security team to silence them. It is quite literally precision medicine on a microscopic scale.
As we wrap up this explainer, I want to leave you with a profound thought from our source material that I think beautifully sums up this whole cellular marvel. Nature designed an elegant system where sometimes silence is powerful. RNA interference gives our cells the unbelievable power to control which genes speak and which genes stay quiet. And honestly, it makes you wonder, if our cells already hold the natural power to intentionally silence the very genes that cause disease, what other incredible secrets are hidden inside our biology, just waiting to be discovered? Thank you so much for exploring this fascinating topic with me. Keep asking questions, keep exploring, and I'll see you on the next explainer.